I'm really excited about what I do, and so I hope to bring that excitement to you today. So the project that I'm going to be telling you about today is, during my time in the lab, I developed a model that I used to answer a basic biological question. And that sounds very vague, because a lot of us work on developing models to answer basic biological questions. But I've been told that this introduction slide is very menacing, so hopefully by the end you will not feel overwhelmed by what I'm talking about. Okay, so we all know, or we all commonly think, that development is a pre-programmed process, that the genetic information we inherit from our parents or our DNA is what helps to contribute to our lifespan all the way through adulthood. However, humans and other organisms alike don't develop in a bubble or in a blank space like you can see on this slide. Instead, they develop in a vibrant and ever-changing environment. And so in order for these organisms to succeed in their environment, they have to be able to interact with these changes that go on around them. And there are several ways that organisms have these interactions and how they overcome them. But one strategy that they use, and the strategy that I'm interested in understanding more about, is that organisms can form bookmarks of their environment. That is, the organisms, when they're young, when they interact with their environment, they can predict that it's very likely they're going to see that environment again in the future. And so they can prepare themselves and respond better to that stress when they see it later on in life or during adulthood. And so to kind of simplify that and make it into a little schematic, the organism early on in life is exposed to a moderate level of stress. Not enough to kill the animal, but enough to suggest, hey, we're probably going to see this again. Let's prepare ourselves. And so this is the point at which an environmental bookmark is made. And so now, later on during life, if they experience that same stress but a more severe version of it, they now have an improved outcome and can survive the stress even if it would be otherwise lethal. These bookmarks come in a lot of different flavors, and some of them are too cool not to show you guys today. So for example, this is a species of grasshopper. These two grasshoppers are genetically identical, and the only difference is that one was raised in a crowded environment and one in an isolated environment. And so this grasshopper raised in the crowded environment knows that it's going to be seen, well, knows, quotation mark, that it's going to be seen, so it develops this warning coloration pattern to keep it from being eaten. While the one in the isolated environment is camouflaged into the leaf because it doesn't need to stand out. So coloration can be a bookmark. Here I'm showing you the water flea, Daphnia. This guy on the right was raised in the presence of predators, so it developed this large helmet and these spiky appendages to help protect itself. But the one on the left didn't have predators around when it was developing, so it didn't have to develop those. And so in this case, the bookmark are these protective features of the animal. Now, if we think about mammals and humans, you don't often see a human developing a helmet early on in life. But instead, a lot of these memories are physiological. You can't see them, but they're just as important for survival. And this is kind of where I'm going to point out why people care so much about environmental bookmarks. And I'm going to use one example, which is hypoxic preconditioning. Now, for those of you unfamiliar with the word hypoxia, this means that oxygen concentrations are much lower than normal. And the reason we care so much about hypoxia is that this is a common feature in heart attacks, in strokes, in severe blood loss. So if we can figure out a way to understand how these hypoxic events are damaging the cells, we can help to protect against them. And this figure at the bottom looks very similar to the first schematic I showed you, in which in hypoxic preconditioning, a moderate exposure to hypoxia can protect against the damage that you see in much more severe hypoxia later on. And so hopefully this example really starts to set up the importance of understanding these types of bookmarks in animals. You can imagine that an organism experiences a lot of different environmental stresses early in life, and so they have a collection of these within their system. And in recent years, actually this year, the White House introduced the Precision Medicine Initiative, which is the idea that we can use the information we know about an individual to specialize their medical treatment. And so if we can understand all of these bookmarks that have either been formed or could be formed in an organism, we can help to determine what kind of medicine we can use to treat patients. And this kind of Time magazine is just highlighting another feature in which your DNA isn't your destiny. It may actually be some of the things that you interact with during your lifetime and not what you've inherited. So hopefully I've convinced you that this is an important question to ask. Why haven't we learned everything we can about it? And some of those reasons is that it's hard to study these type of bookmarks in mammals. Mammals live for a long time, and the type of bookmarks we're thinking about here are bookmarks that can persist over the life of the animal. So to study this in a mouse or any other mammal, it would be expensive and slow going to do. Additionally, when we're in the lab, we want to have a very determined amount of stress given to the animal, and it's hard to deliver environmental stresses to animals since they have respiratory systems and circulatory systems that move the stress around the body. And so we can't really control uniformly the stress in the animal. So let's go back to that original picture that I showed you at the beginning. This is kind of suggesting that what I'm studying is environmental toxins that are released by oil refineries. Maybe. Okay, so I do. Probably didn't suggest that, but I guess that's my head. So the particular toxin that I study is hydrogen sulfide. And as I just told you, this is released at oil refineries. I've spent a lot of my time in graduate school being asked, why do you care about hydrogen sulfide so much? And I could talk for a really long time about that, but for today I want to focus on two main features: that hydrogen sulfide is common and that it's toxic. So I'm already showing you that this gas is released at petroleum refineries. It's also made during the production of plastics, asphalts, at paper mills, and as a byproduct of wastewater treatment plants. So we know that this gas is around a lot. We also know that it's toxic. So if a human is exposed to a high concentration of this gas, it will kill the animal within a few minutes. And we also know that people living around these kind of industrial settings, even when they're exposed to a lower dose of the gas, they have respiratory and neurological defects associated with being exposed to this gas for a long period of time. So this is the gas I'm studying, and you can feel free to ask me later more about why we study it. So I already mentioned that it's hard to study environmental bookmarks in mammals. So instead, we turn to my favorite organism ever, the C. elegans, Centrobditis elegans. So when I tell people I work on worms, they usually picture earthworms, but that's not what I work on. Instead, I work on this guy. And there are a lot of reasons why I work in the nematode. These animals live about 2 to 3 weeks in the lab. And when I mentioned that it takes a long time to study mammals, this is much quicker and we can get our results of these long-term impacts in a shorter period of time. These animals also have been studied quite broadly since the 1970s, so we have a lot of tools, which hopefully I will be pointing those out to you today. And particularly for the things that I study, the way that these worms get gas into their system is important. So a mammal has lungs, but these worms, instead of getting the gas by inhaling it, the gas simply diffuses into the cell. And so we know that every cell within this animal will be exposed to the same concentration of gas as long as we deliver it in a uniform manner. So to deliver the gas, my lab mate, Joe Horstman, and I published a paper a few years ago where we described a new way to expose animals to gases. So hydrogen sulfide, I already mentioned, is toxic. It also smells like rotten eggs, so we want to have it enclosed somewhere where we don't have to breathe it. So here is the fume hood setup that we have in our lab. And if we zoom into it, I'm just going to run you quickly through how we expose these animals to the gas. So we have a large tank of hydrogen sulfide sitting here. We flow that gas through a mass flow controller where we can determine exactly what concentration of hydrogen sulfide the animals will see. We then bubble it through a bubble flask so that we don't dry out the animals, keep the gas nice and hydrated. And then we pump it into this box where the animals are sealed. And since we're pumping this at a continuous rate, the gas flows in and out continually, and the equilibrium will be reached within the box that the animals will always be exposed to a constant concentration. And so this way we can expose our animals to hydrogen sulfide without having to worry about how much gas each animal is getting. So, to summarize this introduction that I've told you so far, there is this phenomenon called epigenetic bookmarking, in which animals exposed to a moderate level of stress can remember that stress even after it's gone, and can ultimately use that to improve their outcome in more severe stresses later on. And this is a hard question to tackle, but what I'm going to be using is a unique new system in which we use C. elegans, hydrogen sulfide, and this fancy fume hood to try to decipher and detect what are the mechanisms by which these bookmarks are persisting. So I already kind of told you that I'm not going to explain to you all the reasons why we study hydrogen sulfide, but why did we think hydrogen sulfide was a good candidate for epigenetic bookmarking model? I'm going to be showing you these schematics throughout the rest of the talk, It's just time from left to right where I'm showing you their embryos over here, and over here they're adults. So like humans, C. elegans die in high concentrations of hydrogen sulfide. So here, if we take an animal and expose it to high H2S for the first time as an adult, we see that all of the animals die within a few hours. So a few years ago, in Mark Roth's lab, Mark Buddy demonstrated that you can precondition animals to hydrogen sulfide. So you can adapt them throughout development in a low concentration of H2S, and then shift them, so this is a sublethal dose, and then shift them from the low concentration to the high concentration, and now these animals can survive otherwise lethal concentrations of the gas that they wouldn't have otherwise. So when I started in the lab, I wanted to narrow down what was the window of time that the animals had to see hydrogen sulfide in order for this preconditioning to work. So the first thing I did was we limited the amount of time directly before challenge, and we found that you can shorten this window to as little as 30 minutes. And even if you only adapt them for 30 minutes and shift them directly into high H2S, they still survive. So it doesn't have to be throughout development, it can be this really short window beforehand. But the thing that I noticed that was the most exciting to us was that you can actually move this acclimation period to very early on in life. So we can acclimate embryos for a very short period of time, I'll go into how long in a minute, and then raise them to adulthood in the absence of the gas. And so now, when they see the gas as an adult, they can survive an otherwise lethal concentration of the gas. And so this memory has persisted for quite a long period of time. So why did we care that this memory can persist this long, and why was it so cool to me anyways? So right here I'm showing you the life cycle of a nematode. They start off as an egg or an embryo, and then they go through 4 larval stages until they become reproductive adults. What we're doing in this experiment is we're acclimating them up here as embryos, and then the animals are going through drastic changes, larval moults, cell division, acquiring the ability to reproduce, and this memory is still persisting over this long period of time. So we were really fascinated about this idea that the bookmark could persist through cell divisions. So I've mentioned it a couple times, but I just wanted to put it up here. The big question that I'm asking in the story I'm telling you today is, how are animals able to remember that they saw a toxic gas really early in life and use it when they're adults? So what is this bookmark? How is it being maintained and persisting? So the best way that I could think of to kind of point out the unique features of hydrogen sulfide bookmarking and why it excited us so much, me so much, is to compare it to some things that are already known. And I've already introduced you to this idea of hypoxic preconditioning. And you can tell that on the outside, hypoxic preconditioning and H2S bookmarking look very similar. We have this moderate adaptation period, a recovery period in the middle, and then you see this improved outcome in high or severe cases. So some work that I don't have time to tell you about the similarities between these two is that I've previously shown in another project that I was working on that you can actually use low hydrogen sulfide to protect against severe hypoxia. And that's been shown in many different situations, not just by myself, but in mice and other tissues, is that you adapt them to low H2S, they can now survive severe hypoxia. So the first question I asked was, can you do the reverse? If you adapt them to moderate hypoxia, can they survive high H2S? And they can't. But this first connection kind of started to suggest to us that there might be some sort of similarities between these two processes. We also know quite a bit about the initial response to each of these stresses, hypoxia and hydrogen sulfide. And interestingly, both of these stresses activate the same protein. So this protein is the HIF1 protein. Now HIF1 stands for hypoxia-inducible factor 1. And the way that HIF1 works is that normally in the presence of oxygen, HIF1 is tagged and sent off for degradation. So when oxygen is present, like in this room, this HIF1 protein is tagged and chewed up. However, some time ago now, we learned that if you take away oxygen, if oxygen goes away, HIF1 is now no longer tagged for degradation, but instead goes into the nucleus of the cells and turns on the genes that are necessary to survive hypoxia. So it's sensed that oxygen is gone, and now we turn on all of these genes to help survive it. A few years ago now, in the Roth lab, Mark Buddy showed that there's another way to turn on HIF1 or keep HIF1 from being degraded. And that is by exposing the animals to hydrogen sulfide. So even when oxygen is present, if an animal is exposed to hydrogen sulfide, HIF1 is not degraded and still goes into the nucleus, but now turns on the genes that are required for survival in H2S. Intriguingly, these genes are not the same. So here I'm showing you a very poorly overlaid Venn diagram because the number of genes that overlap between hypoxia and hydrogen sulfide are very few and far between. But most of the genes that are turned on are completely independent. And the mechanism by which HIF1 turns on these different proteins is still unknown in the two conditions. But so this idea that we're turning on a completely different response in the two conditions was one of the first things that we noticed that was different between these two processes. So let's go back to those schematics, and I'm going to point out a lot more differences that we noticed between the two that really made H2S bookmarking unique. So the first thing is this recovery period in the middle. Actually, in hypoxic preconditioning in worms, this recovery period is required. If you try to move them directly from moderate to severe, they won't be able to survive, or you will still see defects. However, I've already shown you that H2S bookmarking does not require this recovery period. You can shift them directly from low to high. Additionally, the timing of all of these steps is dramatically different between these two processes. Moderate hypoxia, you have to give them at least a 3 to 4 hour dose, while low H2S can be less than an hour. Additionally, the amount of time that these bookmarks can persist is relatively dramatic. Exposure to moderate hypoxia can protect against severe hypoxia about a day later, but no more. And this is in C. elegans. And in C. elegans in H2S bookmarking, instead we see that they can have a bookmark that is maintained for upwards of 4 days, which might not sound that long to you, but if you live approximately 2 to 3 weeks, 4 days is quite a big period of time. So the last feature of H2S bookmarking that's different from a lot of the currently defined bookmarks is that H2S bookmarking is actually reversible. So let me explain. On all the experiments that I've shown you so far, the animals have been maintained on food. C. elegans eat, elegans animals eat E. coli bacteria, and all of these experiments have been done when the animals are fed. But what we found was that if at any point during the experiment you forget to come in and feed your worms, or you take them off of food, you see that the bookmark goes away. And this is relatively rapid and quick. A few hours of fasting will actually make the memory go away pretty rapidly. And this is kind of not a feature that has been seen in C. elegans bookmarks before. So to summarize kind of what I've told you so far in the breakdown of what is H2S bookmarking, I've discovered that H2S bookmarking is a thing, and that adapting animals to low H2S will protect against subsequent exposure to otherwise lethal concentrations later in life. That this is formed very rapidly. It does not require a recovery period. It can persist through rounds of cell division, and it's reversible. So I mentioned in my original introduction that the question I really want to know is, how does H2S bookmarking persist even when the gas isn't around? So this next section of my talk is going to be getting into that question. So we decided to test kind of the simplest or most straightforward hypothesis first, as one does in grad school sometimes. You try to anyways, right? We already know that during this initial response, the HIF1 protein is stabilized. And it's been suggested in the other examples of preconditioning that have previously been identified that this stabilization of this protein, HIF1 protein, is what helps protect against high H2S. So we first asked, okay, well maybe we're just turning on the response and leaving it on. And the response is staying on for all this period of time, and as long as it's still on, it will help out later on in life. Didn't sound too likely, but we're going to go ahead and test that hypothesis anyways. So the tool that we used to test this question, that the response was just simply staying on, is a reporter. And the gene that I'm showing, the gene that we're reporting is SQRD1, which is one of the genes that are activated by HIF1, or turned on by HIF1. So we wanted to see, okay, normally this gene is not on, and what you can see here is we have an animal that has a GFP, or a green fluorescent protein, tag on that gene. And so when the gene isn't turned on, we don't see any glowing in the animal. But when the gene is turned on by putting them in low H2S, we see that the gene is activated and turned on and the animal is green. So if my hypothesis were true and the animal, this activity was being maintained throughout the whole period of the bookmark, we'd expect animals to keep glowing green even after we take them out of low H2S. But we did not find that. So what we found was that when you take the animal out of the gas, the green goes away, or the gene is turned off, which suggests that this hypothesis is not correct, that the response is not simply staying on. So the second hypothesis that we tested, I'm going to kind of take a few steps back. So when I originally introduced environmental bookmarks, we mentioned that the initial response ultimately helps to make an improved response the second time it sees the gas. And what we know about this initial response is that it has to do with transcription, or turning genes on. You have to turn genes on to survive in H2S. So we asked, maybe this improved response is an improved transcriptional response. And so once again, we're going to go back to these glowing worms that suggest when the animals are green, it means that the genes are being turned on. I'm going to show you once again that in the absence of H2S, an animal that's never seen the gas before, these genes are not turned on. So what we did was we took animals that were naive, which means we never tried to form a bookmark of them. They're now adults, and we're going to expose them to H2S for the first time. And you see that the gene is moderately turned on. It's turned on, but nothing too impressive. And what we found was that if we look at animals that were exactly the same age, but the only thing we changed was we adapted them to low H2S early in life, we now see a much stronger transcriptional response to high H2S. And if you don't believe me based on these pictures, we went back and quantified the fluorescence, and you can see that while naive animals do respond to high H2S, bookmark animals respond at a much higher level. And while one gene is cool, we want to know, well, what about all the genes that are turned on in the response to H2S, or all of the initial genes turned on in response to H2S? And to do that, we used a method called quantitative real-time PCR, where we looked at basically the same idea. How much were these genes being turned on? And what I'm showing you are the results of all the top 10 genes, I think this is 10, maybe 9, the top genes that are turned on in response to H2S. And I color-coded this. So the brighter green, that means the better the animal is at turning that gene on if it has a bookmark. And so you can see, for example, this gene up here, GST19, is turned on 181-fold higher in bookmarked animals than naive animals. So going back to our model, we can now add a new piece. Not only does this bookmark improve survival in high H2S, but it also leads to a robust transcriptional reactivation of the genes that are required for surviving in H2S. So the only kind of real piece here so far that we're missing, we know what the initial response is. We know that they survive. We have demonstrated this. What about this bookmark? How is this bookmark being formed and maintained? Thankfully, I work in Elegans, and this is we can use a genetic screen to try to determine what genes or things are required for H2S bookmarking. So all of the experiments I've shown you so far are in wild-type animals, or animals without defects in their genes. What we can do, and what people have done in the past, is you can induce mutations in the animal, either through directed or random mutagenesis. And what this ultimately ends up allowing you to have is a collection of animals with mutations in single genes. So for example, this animal here has a defect in one particular gene, while these animals have defects in other genes. And so we have a large collection of these in nematodes that we can go to and try to ask questions about whether the gene is required for function. So I'm going to give you a quick hypothetical description of how this experiment works. So the question I'm asking is this animal able to maintain a bookmark of H2S? I've already shown you that wild-type animals, yes, they can. And for most of these genes that we're going to screen, most of these animals that we're going to screen, the gene that we knocked out is not required for the bookmark. And so most of them will form a bookmark and maintain a bookmark just fine. But if we happen to have made a defect in a gene that's required for H2S bookmarking, they won't be able to form the bookmark, and you'll see that they'll die in high H2S. And so based on the results of this screen, we can conclude that gene X up here is the gene that's required for H2S bookmarking. So I did this. And what we did was we sat down and thought up very good guesses for what H2S bookmarking could be maintained through. And here I'm showing you a few of the categories of the guesses that we had. But we ended up compiling about 350 candidate strains, so genes with defects in over 350 genes. And we wanted to see if they could form a bookmark of H2S. So we did this in a two-part process. The first step was that we acclimated the animals to low H2S and then looked to see if they could survive high H2S. And as I mentioned in my hypothetical screen, we were looking for animals that couldn't form a bookmark, maintain a bookmark. So we looked for much less than 100% survival. Now you can imagine if you look at this, that you might pull things out of here that you weren't looking for. For example, animals that have defects in just responding to H2S alone would die in this process. So in order to kind of narrow down really and focus on what we wanted to understand, we added a second step to this screen. So any animals that had less than 100% survival here, we did a second experiment where we took them, acclimated them to low H2S, and then shifted them directly to high H2S. And this way, we wanted to see that all of these animals were surviving. And so the difference between these two steps is this middle period. And since the question I'm asking is what genes are required for H2S bookmarking to be maintained, we've now really narrowed it down to animals that have defects just in this step right here. So from this screen, we identified 7 candidates, and I'm going to talk about quite a few of them today. And as a little bit of a spoiler, all of the candidates that we found fall in this category of epigenetic factors. And so now we're going to get into what were the candidates I found, what do they do, and how do we think they're working in H2S bookmarking. So the DNA within our body is organized to allow certain genes to be turned on and off when we need them to. And one of the more basic levels of DNA organization is they are organized into nucleosomes. So this red string here is DNA. And it's wrapped around a set of 8 histone proteins. And this whole thing right here is known as a nucleosome. And what you can see is that the DNA that's wrapped tightly around these nucleosomes is hard to get to if you need to turn them on, but the ones that aren't associated with them are easier to activate. And so these nucleosomes are placed in a non-random manner throughout our genome. And I'm going to focus on 2 particular states of nucleosome organization. So in some cases, the nucleosomes are spaced pretty far apart, and you have all this DNA that's accessible. And this is known as an open chromatin state. So several nucleosomes along DNA is known as chromatin. And so we can see that this gene can be activated. It's transcriptionally possible because we have access, we can bring in all the machinery we need. However, there's also closed chromatin, and this is when the nucleosomes are densely packed together. And now you can't get in there and turn that gene on. So this impedes transcription or gene activation. So to give you a quick summary of the candidates that I'm going to talk to you about today, they all play a role in switching back and forth between these 2 states. And I'm going to go through them each one by one, but what I just want you to see is that they are all really involved in this switching back and forth between open and closed chromatin. So my first section is going to be on histone modifiers, and I'll tell you what that means in a minute, but I first want to show you what our data from our original genetic screen looked like. So I already mentioned that wild-type animals are able to form a bookmark of H2S just fine. So here on this graph, I'm showing you the percent of animals that survive high H2S, and we see that wild-type animals survive just fine. However, animals with mutations in 2 genes, SET2 and SPR5, have dramatically reduced survival, even if bookmark has been formed before. And as I mentioned in the second part of our screen, we wanted to make sure that these animals didn't have any defects in responding to low H2S. And we saw that much like wild type, these animals would survive just fine if we shifted them directly from low to high H2S. So what are these 2 genes? What is SET2 and what is SPR5? So when I showed you the original picture of a nucleosome, which has DNA wrapped around 8 core histone proteins, I didn't point out that each of these histones have long tails hanging off of them. And to regulate gene activity, or how a gene is turned on, there are modifications that can be made. So here I'm showing you the addition of a methyl group or an acetyl group, and these marks can be added to help say, hey, turn this gene on, or hey, turn this gene off. And both SET2 and SPR5 work on the same tail, at the same place on the tail, of the H3 histone protein. Now, to of course make things even more complicated, this part of the tail can be changed in several different ways. So here I'm showing you the H3 protein. We're tagging it at lysine 4 or K4. And it can either be mono, di, or trimethylated at this site. And there's different machinery that helps convert between these steps along the way. So I already mentioned that SPR5 was one of the genes we found. And SPR5 is known to function in the co-rest-like complex, which helps to take off one of these methyl groups from the dimethyl and form the monomethyl. Now, since I said complex, this means that there are several proteins that work together in this step. And so what I did is I went back and found animals with mutations in each of the members of this complex. And what we found was that not only does SPR5, the thing we identified in our screen, but the other 3 genes that are involved in the co-rest complex also had defects in H2S bookmarking, which at least genetically suggests that this step right here is really important and that it is functioning as the co-rest complex. Now, set2, the second gene we identified in our screen, is a little bit more complicated. So we know that it plays a role in at least 2 steps along this pathway. Not only in the addition of a methyl to make dimethyl H3K4, and it also functions as a member of the ASH2 complex in making H3K4 trimethylated. So at least to start to get at this question, we wanted to look at members of each step. So I already mentioned to you that set2 alone has a defect in H2S bookmarking. But we wanted to look at other members of the ASH2 complex. And in contrast, we found that the members of the ASH2 complex that we looked at did not have a defect in H2S bookmarking, suggesting that it is not working as a member of the ASH2 complex, but instead over here at this step. And to kind of make myself feel a little bit better, at least genetically, we also looked at RBR2, which functions antagonistically of ASH2 to go from tri to dimethyl. And much like the ASH2 complex, we also found that these animals did not have defects in H2S bookmarking, which suggests that it is the co-rest-like complex and the set2 protein that are required for H2S bookmarking, and that there may be something important in mono and dimethylated H3K4. So going back to the model that I've been building for you from the beginning, we can now add that this bookmark may very likely be due to regulation of H3K4 mono and dimethylation. I guess one thing I forgot to point out is that it's kind of crazy that they're working in opposing functions, but both giving us the same phenotype. And you can think of several reasons why this might be happening. They could be working at different target sites, so set2 is working to turn some genes on, and co-rest is functioning to turn some genes off. Or they could be regulating the same sites, and you just have to have a nice kind of perfect window of methylation. So those are 2 possibilities that this might suggest. So what I've shown you so far is that H3K4 di and monomethylation is required for, well, I haven't shown you that, but is required for survival. So the next question we wanted to ask was, is it also required for this transcriptional reactivation that we see? And so I'm going to turn back to those quantitative RT-PCR experiments I mentioned earlier. And just as a reminder, what we're looking at is how highly is this gene turned on in bookmarked animals compared to unbookmarked animals. So I already mentioned that wild-type animals, you see that these genes are turned on at a much higher level in bookmarked animals. And we do the exact same experiment in set2 and SPR5 animals that have defects in these 2 proteins. You see that there's a lot less green up there. There's a lot more white. And so what we see is that all of this robust reactivation is dramatically reduced. In some cases all the way, but in some cases just much less. Which suggests that set2 and SPR5 are functioning in controlling the transcriptional response in bookmarked animals. So I mentioned that there were going to be kind of 2 stories that I was several candidates that I was talking about. And so I've taken some time to describe SPR5 and set2, which both function to change the methylation status at histones. But the second story I'm going to tell you about today is about the switch-SNF complex, which functions in a different role, but at very similar locations, in that it helps to pick up and move around nucleosomes. So instead of just tagging them to say whether they should be turned on or off, the the switch-NIF complex pushes them around or takes them off completely. So the reason that we suspected the switch-NIF complex was that in our initial screen we saw 3 independent members of the complex be pulled out. So here, these are 3 animals that have mutations in different subunits of the switch-NIF complex, and all of them had trouble forming and maintaining a bookmark of H2S compared to wild-type animals, which do just fine. So much like the co-rest complex, the switch-NIF complex has many, many subunits, and when we went back and pulled out animals that had defects in each of the subunits of the complex, we saw that the pattern continued, that animals with defects in any subunit of the complex had, and to varying degrees, defects in H2S bookmarking. So once again, we can suggest this tells us that the switch-NIF complex is functioning as a complex in H2S bookmarking. So much like the animals that we looked at in the histone methylation story, we next wanted to know if these genes were just required for the survival that we saw, or if they were also required for that transcriptional reactivation that we usually see associated with it. So now I'm going to add one last panel onto this graph. We already show that SPR5 and SET2 animals have reduced transcriptional response, but we found that animals with mutations in the switch-NIF complex also do. So once again, much less green. These animals aren't able to turn these genes on as well. So going back to this picture, we've now identified 2 complexes and a histone methyltransferase that are involved in H2S bookmarking. And we can start to form models, and I'm going to get there in a second, of what we think is going on and how these bookmarks are maintained. But the transcriptional response that I showed you was all at H2S inducible genes. And so we would expect that if these genes that are normally turned on in H2S are modified by these complexes, we would find them sitting there at some point along the way. And to do that, we can kind of take advantage of some cool tricks that we have in biochemistry to identify whether these guys are actually sitting there when we think they are. Are they actually in the place that I think they are at the time that I think they should be? And so to do that, we can basically stick the switch-NIF complex to a location that it's being bound to. And so I imagine that we have an animal, we've exposed it to H2S, we're forming a bookmark. Switch-NIF is probably going to be sitting right at these genes that I think are being activated in H2S. And so what we can do is stick these 2 together, and then we can sequence the DNA that's associated to this, do a chromatin immunoprecipitation, PCR, and see whether this guy is sitting at the DNA that we think it is. And we found that it indeed is enriched in bookmarked animals. So here I'm showing you 2 examples of H2S inducible genes, or genes that are turned on in H2S. And we expect it to be sitting in the promoter of the gene. Now I didn't take too much time to describe this, but the promoter is where kind of the regulation of the gene activity happens. This is before the gene starts. And what I found was that we saw more of the protein, more switch-NIF, sitting in the promoter of this gene than in places we wouldn't expect, like the coding region or at other sites that are not induced by H2S. And so this y-axis here is showing you fold enrichment. So we see that after we formed a bookmark, the switch-NIF complex is sitting there at a much higher level than it would before we formed a bookmark. And this is the same in this other gene that I'm showing you. Once again, more switch-NIF complex sitting specifically at these genes after we formed a bookmark. So, mostly wrapping up, let's form a model. I've shown you a lot of information today. Let's make a model about what's going on. So the pictures that I'm going to be showing you are all going to be the promoters of H2S inducible genes, right where we're controlling whether a gene is on or off. So before an animal ever sees H2S, the chromatin is in a closed or off state. It's not being turned on because we haven't activated the response. The gene is off. When we expose an animal to low H2S for the first time, the gene needs to be turned on. I have it, yeah. Needs to be turned on. The HIF1 protein needs to come turn this gene on along with other transcriptional machinery. And we're suggesting that during this initial exposure, this is when the switch-NIF complex and these other genes are recruited to the location, the promoter. And so now, when the animal is removed from H2S and the gas is no longer there, HIF1 is going to go away. So we're not going to turn on this gene, but we're going to maintain this open or primed state. So the gene's just going to stay open in anticipation that it may have to be turned on later on. And this is done by the addition of these marks, the remodeling of the nucleosomes, location. And so now, this animal, after a period of time, is now exposed to a whole lot of H2S. And so now, since we have this primed chromatin, we're already ready to go. HIF1, the transcriptional machinery, can come in, and these genes can be turned on right away. And we can get this activation to a really high level at this step in the process. So to summarize the information I've told you today, I mentioned that environmental bookmarks are important, and what they are that you are able to survive severe stress if you've seen the stress before and can be prepared to respond to it. This is hard to study in mammals due to length and expenses and a lot of reasons. But I've shown that the same type of bookmarks are formed in worms, specifically to hydrogen sulfide, where we can adapt them to low and now see they survive high H2S. I've shown that not only do we see that these animals can now survive otherwise lethal concentrations, but they have this improved transcriptional response, which may be what is allowing the animals to survive in high H2S. I've then gone on to show that we can really get into the intricacies of how this works, taking advantage of the genetic power of C. elegans. And I've shown you that there are several complexes, the switch-NIF complex, the co-RES-like complex, and the SET2 protein, that are required for the formation of this bookmark. So after telling you all of that, why is this cool? Why is this important? I mentioned earlier this idea of the precision medicine initiative, but you can imagine that if we are able to understand how these bookmarks last for a really long time in organisms, we can utilize that information to our advantage whenever we need to know it, like if we're treating a disease or aging. You can also very quickly imagine that if you tell someone who works in agriculture that you can help improve the stress response in their in their plants, they're going to be happy to hear that. On the sides of hydrogen sulfide, OSHA currently doesn't take epigenetic influences into account when they are setting their health and safety standards for environmental toxins. And hopefully, at least somewhat today, I've shown you that epigenetics can be changed by environmental toxins, and that we probably should have a closer look at how these toxins are changing our epigenome, which I never defined the word epigenetics. It's what I've shown you today. So, wrapping up, the things that organisms encounter early in life are really important and can have dramatic impacts on the adult animal. And I've demonstrated that this can occur with the environmental toxin hydrogen sulfide. One thing that I didn't point out is that all of those complexes that I pulled out in my screen are conserved in mammals and plants and lots of other things. In fact, bookmarks in plants are kept and maintained by very, very similar mechanisms and the similar complexes that I found. So this really suggests that there's some conserved mechanisms throughout a lot of organisms that may all use kind of the same mechanisms to form and maintain these bookmarks to improve survival later on. So with that, before I get into my acknowledgments, since I am a crier, I want to take your questions, and then I ask you to please stick around for a few minutes so that I can cry.
Question. I just want to say, before anyone has a chance to escape, that there will be, as Alex Murs so eloquently put it, a celebration or wake after the defense is over in the lounge between the J4 K3 wings. So that's just down the hall from the biochem office. Okay, now questions. Go.
Okay, Laura first.
I have a lot of questions. Really awesome, compelling stuff. The first is, so given that these are epigenetic changes, have you looked into how heritable they are by subsequent generations?
I have, and I really wanted them to be heritable, but they're not. We found, and Dana spent some time looking at this as well, it's lost at meiosis. So it really, we saw no changes at all in offspring, sadly.
And then when you were looking at, I mean, these are pretty generalizable phenomena, right? Chromatin remodeling, response to stress. So could you speculate a little bit about like what specifically is changing when you're fasting the worms and why that goes away?
So that is like my favorite pet project, and I would love to get someone to work on that in the future. I'm going to recruit really hard this year to get a graduate student to work on that. So there's a lot of ways that fasting could be reversing the bookmark. Kind of, I don't want to say boring, but less exciting thing is it's just an indirect effect. Fasting also changes chromatin, and it might just be pushing around or recruiting factors to one place over the other. I did a small kind of biased screen looking at all of the responses to fasting, and I couldn't find anything actually that was required that would have, the fasting response that would have affected it, but I'm excited to look into that. Yeah?
Yes, that was a beautiful presentation.
Thank
I've been
you.
around for 50 years, one of the best I've heard.
Thank you.
I had a couple of questions. Well, I'm very interested in the variance. I was standard errors and lots of variance.
There is.
I was wondering, any experience in the variance as a function of the age of the C. elegans? Do they have more heterogeneity? Maybe
Yeah.
some stochastic events?
Yeah, so I have done some work on, as they get older, do we see more of a variation? With these the methylation defects are the highest variance I see in any of the things that I do. So I think it's very specific to the methylation. There was a bit in those, but the switch-NIF ones are a little bit condensed. They get worse at maintaining the bookmark in general as you do it in older and older animals. So that already starts to introduce variation, just because, you know, after day 5 or 6 of adulthood, it really starts to go down on whether they can maintain it at all.
A quick other question. Do you think they're paying a price, or we're paying a price from early exposure and setting us up because you have a lot of open DNA, maybe a lot of more DNA damage?
Yeah, so I've definitely thought about that kind of stuff as well. I don't think there's huge evidence for that. So sometimes if you make a bookmark and you predict wrong, there can be pretty dramatic consequences. The idea if a mother, the baby in its womb, if the mother, you know, it doesn't have good nutrition and the baby is then born into America where they can eat cheeseburgers all the time, they have problems. So there's definitely some trade-offs. We haven't found them yet, but the fact that fasting can reverse it and it's so readily reversible makes me think there might be some trade-offs. Yeah. Joe.
So, if I'm correct, that you have the two methyltransferases, they're acting like, you know, opposing ways that have the same or similar phenotypes. Can you create the double mutant and see what phenotype that would have?
Yes, I would love to do that.
What do you predict it would be?
So I really don't have a good feel yet on whether they're working in the same place or different places. We can definitely push methylation in one direction or the other and see if we can, if it is this small window, if we can push ourselves outside of the window in either direction. I don't know exactly what I would expect to see. We've had the idea that if you knock them both out and then they're just fine, that would be awesome. Two bad things make a good thing, which could, hard to speculate. Irini back there.
Thanks very much for the good presentation. I have two questions. Probably I missed the part about the first question. Did you do a chromatin immunoprecipitation during the recovery state to see that splicing is still attached to the promoter?
I would love to do that. I haven't done it yet. So, yeah. So the question is, the chips. Have we done the chromatin immunoprecipitations all the way through the process? Is it being stuck there? Is it setting up the chromatin and leaving it there and going away? That's next on my list, and we've been working on it, but I haven't gotten there yet.
And the second question is, did you by any chance, because you noticed, you said at some point that when you pre-expose the animals in low hydrogen sulfide, you see that they can survive in hypoxia. Did you check by any chance to see if SNF binds to the promoters that are that are induced during hypoxia?
Yeah, no, I haven't. So,
Put them in low hydrogen sulfide.
right. I haven't looked at switch-NIF and hypoxia yet. So hypoxia does recruit switch-NIF to places. It's been shown previously that switch-NIF is involved in hypoxic activation.
I expressed the question very wrongly.
Sorry, okay.
It's my fault. I mean that when you check if Swiss-NIF,
Oh, does it go to hypoxic targets?
doesn't go to hypoxia targets.
I haven't looked at that yet. It would be interesting to see if even though HIF1 is only turning on a subset of genes in H2S, is it recruiting things to other places?
That could explain that they still survive in hypoxia.
Yeah. So the hypoxic preconditioning story is a bit complicated. Some people think that hypoxic preconditioning itself turns on some endogenous H2S, and that H2S itself is part of hypoxic preconditioning. So I don't think it's going to be as simple as just recruiting switch-NIF, but it's something to look at. Michael.
So I think of these chromatin modifications as being very global. So how do you get the specificity that just these genes are the ones that are specifically turned on? Isn't switch-NIF acting at huge numbers of?
Absolutely. So the switch-NIF complex is working all over the place.
And the same with the methylation.
Mm-hmm. So I went through and I was really shocked that it was so specific. I went through and looked at hypoxic targets. I looked at the transcription of a bunch of different genes and didn't see anything change except specifically H2S targets. And so that kind of gets back to this idea that we don't know why HIF1 turns on a certain set of genes in hypoxia and H2S, but it presumably suggests that there are other cofactors involved. And it may be the recruitment by those cofactors and HIF1 that is specifically bringing it to the sites that they need it.
So another side question is that I noticed that in any of your single mutants, you were still having some survival.
Mm-hmm.
So is it that each of these is it all transcriptional, or have you ever made any double combinations, or have you ever been able to completely eliminate it, or is there some non-transcriptional part to that?
So I spent a really long time, like my second year of grad school, trying to do RNAi experiments to make doubles. But we found that, I already mentioned that fasting reverses H2S memory. Well, if you change the food source at all, even switching to RNAi food, it completely wreaks havoc on the system. So I spent a really long time doing that, and it didn't work. I've looked at some doubles, and I think a lot of these mutations are really bad for the animal because they're used in a lot of different steps along the way. Not all of them, but some of them. So I think some of that residual survival is probably because they're not complete nulls in some of the cases. But in other cases, I think it might be that they're working in concert and having, if I knocked all of them out, I think I would see a stronger defect, but I haven't done it yet. Steve.
You mentioned that the bookmarking was reversible with changing the diet. Is it reacquirable after it's already been forgotten?
I still haven't done that experiment. I don't think I did it. Oh, well, she did it, and it works. That's always been, I wanted to know how many times you could reverse it and put it back, but I don't think I've ever done that.
Is there a decrease in performance in the second time, or?
I didn't do it that way.
Yeah, I know, that's a great question. I don't know. Billy, did you have your hand up?
Just kind of quickly, do silicons molt at all? Do they shed, do they alter the way that they might be absorbing H2S?
Wait, what?
Do they go through some kind of shedding?
Yeah, so there is a larval molt between the larval stages.
And then even as an adult, is there another one?
I don't think so. So you were thinking that they were...
How is that stage affecting absorption?
Oh, so I could, what I kind of left out of that talk was I can adapt them as embryos, but I can also adapt them anytime, ever. All the way through larval development. So it's not specific.
All right,
Sorry.
the party is never going to get started. No, just in order to keep us all on.
Oh wait, I have acknowledgements.
Yes, that's what
Okay.
I'm saying. Now. And then more questions with the liquor.